Good day everyone, I'm Sir Franz and welcome to our lesson on monoclonal antibodies, and in particular we'll be talking about the basic principles and why these techniques are used and how monoclonal antibodies are synthesized. So first, monoclonal antibodies are derived from a single activated B lymphocyte. If you will recall, there are a lot of B lymphocytes specifically activated for unique antigens and pathogens in the body. Monoclonal antibodies, therefore, will mean antibodies that are derived from a single line of B lymphocytes. They specifically act on a particular introduced antigen, and they are produced by fusing the B lymphocyte with a cancer cell, which is inherently immortal and therefore will confer that immortality to the B lymphocyte, allowing continued antibody production. So first we have to understand the architecture of the spleen. The spleen is an intraperitoneal organ, which means it's an organ inside the abdominal cavity. In particular, it is behind the stomach and in front of the left kidney. So the spleen, if you will recall your anatomy and physiology, is important for producing antibodies and protecting us from encapsulated bacteria. It is also important to remove the effete or old red blood cells from the circulation. So let's take a closer look into the architecture of the spleen. This is the fibrous capsule of the spleen, which introduces septations or trabecula into the organ. And these are vascular sinusoids, which, if you think about it, are just enlarged blood vessels, and blood will flow here, and red blood cells will have to go out of the sinusoid into the red pulp, where they will stay and they will have the chance to be removed if they're already old. Next, you have white pulp, which is here in the middle. And in the white pulp, you will see a primary follicle, which is composed of a lot of lymphocytes and macrophages. We have the marginal zone, which is at the periphery, and you have periarteriolar lymphoid sheath, which are collections of lymphocytes around blood vessels, in this case an arteriole. And what's unique about the spleen is that it has germinal centers, where you have lots and highly active proliferation of lymphocytes. Then this is your splenic artery and vein. So what's important for us in our discussion of monoclonal antibodies would be the white pulp, and in particular, the germinal centers. Again, collections of lymphocytes that are actively proliferating. Next, let's just recall how antibodies are formed. First, you have professional antigen-presenting cells, which are in the form of macrophages or dendritic cells. So they will engulf the pathogen or the antigen and process that antigen, associate it with MHC, major histocompatibility complex, and this is your antigen. And what this does is it literally presents the antigen as if it is food in a silver plate. And by doing so, it will allow the helper T cells of the body to uniquely recognize the antigen. If it has already encountered the antigen, ibig sabihin nun, presentation has occurred, the T cell will become activated. And once the T cell is already activated, it will further activate a B cell directly and by secreting cytokines, which are messengers that travel in the blood. And its effect would be the proliferation of B cells. So let's recap that. You have your antigen presenting cell taking up a pathogen or antigen, presenting that antigen using the major histocompatibility complex, allowing the helper T cell to recognize the antigen. After recognizing the antigen, it will become activated and will further activate another class of lymphocyte, the B cell, which further differentiates into antibody secreting plasma cells. And these plasma cells will be the ones that produce the antibodies that are specific to the presented antigen. So how does activation of the B cell into a plasma cell occur? By direct activation using helper T cells. So mag-uusap sila directly, or by the secretion of the helper T cell, which are cytokines. So at the end of the day, you will have an activated plasma cell which produces antibodies. So let's now go into the meat of the discussion. How do we produce monoclonal antibodies? So again, let's remember that monoclonal antibodies come from only a single specific lymphocyte. So how do we do it? We immunize mouse or a rabbit with a particular antigen. So we inject that antigen and we hope that the immune system of the rabbit recognizes the antigen. After the antigen has caused activation of a particular subset of lymphocytes, you will have antibody forming cells, plasma cells, from the spleen. You get the plasma cell which is specific for that antigen, and you combine it with a cancer cell right here. So you grow simultaneously a culture of cancer cells, which are usually activated lymphomas. So we fuse the two, the antibody producing cell, plasma cell from the spleen and the cancer cell. So you fuse the two and you form a hybridoma. So this hybridoma is important because it has the two properties that we want from the antibody producing cell from the spleen and the cancer cell. So we have the hybridoma producing antibodies that are specific to the antigen that we introduced, plus the hybridoma, because it contains material from cancer cells, will also express immortality. So as long as the medium, which is nutrient rich, is replenished, the hybridoma will continue to divide and it will continue to grow because it's cancer cell, but at the same time, it will continue to produce that specific antibody that we are concerned about. So the hybridomas are screened for antibody production. We have to check if they produce antibodies. And then we subculture it. We remove a cell and implant the cell on a lot of petri dishes. Hopefully, these hybridomas will produce the antibodies that we desire. And we purify the antibodies and package it into a medication. So at this point in time, we have already discussed monoclonal antibody production. And we have lots of monoclonal antibodies. For example, the monoclonal antibodies that we recently used to help us manage patients who are severely afflicted with COVID-19 infection. For example, you have monoclonal antibodies against interleukin-6. You have monoclonal antibodies against interleukin-1, monoclonal antibodies against tumor necrosis factor, and you can even have monoclonal antibodies used to treat cancer. For example, bevacizumab, which is a monoclonal antibody specific for preventing neovascularization of tumor cells. So let's have a quick recap. We know that antibodies are specific proteins that originally are produced to combat infection, but there are also antibodies that are produced to combat specific pathophysiologic processes, for example, cancer, autoimmunity. We produce monoclonal antibodies by selecting a particular activated lymphocyte and then combine it with a cancer cell. And hopefully, that cell lineage, which we call the hybridoma, is monoclonal, but at the same time, it is immortal. And we just continuously provide nutrients for the hybridoma, and we collect the products of the hybridoma, which is the antibody. So with that, thank you for listening.